Hi, so today we are going to be discussing the major steps in oogenesis. And we're going to start at the stage of the primordial germ cell and then work our way until fertilization and extrusion of the second polar body. In this video, we will be focusing mostly on when is mitosis occurring, when is meiosis occurring, how many complements of DNA is in the cell at each point, and when is meiosis arrested and lifted. So before we get too far into this, I want to just go over the 4 major phases of maturation that an oocyte must undergo. The first step, or the first phase, is that primordial germ cells must divide by mitosis twice. These happen before the female is even born and are known as the prenatal steps. The second phase is meiotic arrest occurs in the primary oocyte's nucleus. This first meiotic arrest, or nuclear arrest, occurs at prophase 1. We'll have another bout of nuclear arrest at metaphase 2. The third phase is that the cytoplasm of the primary oocyte grows. Cytoplasm of the primary, I'm putting a 1 prime here for shorthand, oocyte grows. This is phase 3. And the fourth phase is that meiosis can resume again after the pre-ovulatory LH surge. Now, know that when the LH surge occurs, that doesn't mean that all of the primordial germ cells are reaching this same phase. This is what happens to one oocyte at a time, because you're going to ovulate one at a time. And I'm going to write shorthand here, pre-ovulatory, pre-ov. You might see me writing that again later. And LH, by that we mean luteinizing hormone. If you want to know more about the action of luteinizing hormone, you can check out some more videos that'll be in the hormone series. So we start out here with a primordial germ cell. These primordial germ cells actually, during embryonic development, they originate from the yolk sac and they migrate to the gonadal ridge. Now, in this cell that I've just drawn, I want to explain what's being represented here. So, this is a cell. This is the cytoplasm. This is the nucleus. In this nucleus, and obviously we're not representing every chromosome here, but in this nucleus we have this is a diploid cell, and as I've drawn it here, it is 2C. Now, what's going to happen, and also, if the thought of complements, that's what 2C means. 2N means that there is 2 sets of DNA present here. 2C, that's just referring to individual chromatids in the nucleus. Now, what's going to occur is that this primordial germ cell is going to undergo mitosis. So its DNA has to replicate. This will give us and so now this cell is still 2N. It is still diploid because we haven't done anything about the content of the DNA. We haven't done anything about the genetic content of this cell other than there are more copies now. So now this is 4C. And you can look, as I've drawn these arms, these chromatids, they're kind of C-shaped, if that helps you. So this is undergoing mitosis, and what's going to occur next is that our result will be cell division as well. So we are going to, from this one cell, from our previous slide, end up with 2 oogonia. These oogonia will have in each of them they are exactly what was in the previous, they're an exact replicate of that primordial germ cell. And don't be confused here, the way that the chromatids are drawn, they are not connected. These are not sister chromatids. It is just a replication of the primordial germ cell, and these are known as oogonia. These oogonia are going to undergo mitosis again. So the same process of DNA replication where we are now going to have, I'm not going to draw the DNA replication again, but our result will be 4 primary oocytes. So from the oogonia, we get 4 primary oocytes. These oocytes are genetically identical to those oogonia, which was genetically identical to the primordial germ cell. Each of these primary oocytes are 2N, because there are 2 sets of DNA, and they are 2C. We have 2 little C shapes, 2 chromatids. Now, what's going to happen next is meiosis. So these primary oocytes, and I'm just going to represent it with 1 right now. So if we look at a primary oocyte, the next step is meiosis. So what we are going to get is this. Because we're going to have to replicate this DNA. So, just to be clear, this primary oocyte is still 2N because we haven't changed the genetic content aside from the fact that there are more copies now. So this is 2N and 4C. We have 4 chromatids represented here. Now, this isn't going to continue through all of meiosis because the primary oocyte will enter the first meiotic arrest after meiosis prophase 1. So, this primary oocyte right here will be under nuclear arrest, which means the further steps in meiosis cannot occur. We can think of the nucleus as kind of frozen. It's like a factory line, and everyone has put their hands up in the air. Nothing else is happening. And the formal term for this nuclear arrest is dictyate. This primary oocyte will remain in nuclear arrest until an LH surge. And the LH surge that we're really talking about here is the first one that the female animal will experience, which is when she enters puberty. So again, that doesn't mean that all of the oocytes will have their meiotic arrest lifted at the first LH surge of puberty. It just means that the first oocyte to be ovulated will be lifted from meiotic arrest when puberty begins. So this is obviously going to continue throughout the female animal's life, so she's not going to just dump out all of the eggs at once. While under nuclear arrest, something else is happening to this primary oocyte. So we can't forget from previously from one of the major phases of oogenesis is we need cytoplasmic growth, and we need to form a Zona pellucida. So this is what the oocyte will look like once it has a Zona pellucida. So we're going to have, I'm going to represent the Zona pellucida with this thick blue line, and the zona is kind of analogous to the shell on an egg. It is a very thick jelly-like layer, actually made up of mucopolysaccharides that are synthesized by the oocyte itself. So this is a protective layer, the Zona pellucida. The sperm will have to penetrate this, and we'll discuss that reaction in another video. But eventually, the oocyte will look a bit more like this. So, again, there is our cytoplasm. There's our Zona pellucida. This space here is called the perivitelin space. The perivitelin space. This is the space into which polar bodies will be extruded in subsequent steps. So, while under nuclear arrest, the oocyte experiences cytoplasmic growth and formation of a Zona pellucida. Also involved will be the formation of gap junctions, but we are going to discuss that more in a video about the placement and lifting of meiotic arrest, so you can hear about that in more detail later. So the zona, like I said, is a jelly-like layer encapsulating the oocyte. And next, we're going to say now that an LH surge has occurred. So the LH surge, luteinizing hormone coming from the anterior lobe of the pituitary, gives us this. I'm going to again represent the zona with a thick blue line. And there's Zona, pellucida. This is the perivitillin space between the zona and the oocyte itself. And this perivitillin space is where the polar body will be extruded. So you might ask, okay, well, what's a polar body? After the LH surge has lifted meiotic arrest, this secondary oocyte, I'm going to denote that with a 2', will complete meiosis 1. After completion of meiosis 1, here's what the cell's nucleus will look like. Because remember, before we had a 4C cell. So this is a replicated chromosome. You see here 2 sister chromatids. And the amount of genetic material that's found in the oocyte, in the secondary oocyte at this point, is the same that will be found in the polar body at whatever stage. So here, in our first polar body, we have the same amount of genetic material. So the polar body is the oocyte's way of getting rid of that extra genetic material to increase offspring survival, because obviously too much genetic material will lead to unfavorable mutations and prevent survival. So instead of the cell dividing into 2 cells, it just extrudes this polar body. This is polar body 1. And the secondary oocyte and the first polar body both can be said to be 1N, meaning it is haploid, because we have changed the amount of choices for genetic material here. And since there are 2 chromatids, we say that each is 2C. Now this is going to experience meiosis again. So it's going to go into meiosis 2. And at meiosis 2, this secondary oocyte the result will be an otid. However, it's going to enter a second meiotic arrest at metaphase 2. And to be clear here, this is a secondary oocyte. It will begin meiosis 2 and will enter nuclear arrest, just like we explained before, at metaphase 2 this time. While it's arrested at metaphase 2, this will ovulate. This oocyte, well, it's on its way to becoming an otid at this point, will ovulate, and you'll have this as the result. So we have here now, there's polar body 1. And whereas previously This secondary oocyte was in and 2C. The sperm comes along. And this sperm contains in its nucleus, it is in, one in, one C. I'm drawing 1C in there. A lot of these events happen very quickly, so when fertilization occurs, the meiotic arrest, which had this otid frozen, metaphorically frozen at metaphase 2, will be lifted. When that happens, we're going to have a nucleus like this in the oocyte. The second polar body will be extruded. The second polar body will have just as much information in it, just as much genetic information as the oocyte's nucleus. So there's our polar body 2. That's extruded right after fertilization, right after this sperm binds to the zona. And here we have 1 in, 1 C. And after that, you're going to start to see a breakdown of the nuclear membrane, the nuclear envelope, so that this will actually start to look more like that. So our next step is, what does this look like after fertilization? Previously we had an otid. That was what had otid was 1 in, 1 C. Fertilized by a 1 in, 1 C sperm. And our result is this, the zygote. Zona pellucida is still in place. It's a protective layer. Only one little part was invaded by the sperm. And we have here the extruded polar body 1 and 2, which will go away. And now we have pronuclei have formed. The male and the female pronucleus. That is directly after fertilization. And the subsequent events for the zygote and activation of the offspring's genetic material, that will be explained in later videos. I hope this has helped you to better understand a primordial germ cells, so to speak, journey and the processes it goes through, including different stages of mitosis and different moments of nuclear arrest on its way to become a zygote. Thanks for watching. Keep up with the series.